Okay. In the interest of time, I might just jump right in. I know we're a minute early, but I just got some housekeeping stuff to walk through anyway. And hopefully by the time I get through it all, we'll have full time ready for our presenters. So thanks everyone for joining us today. Welcome to the Collaborative Science for Estuary series. This is Nick Sobral with the Science Collaborative. Today's session features a collaborative research project that brought together a network of intended users from reserves, state agencies, and Chesapeake Bay nonprofits to develop a framework to guide eelgrass restoration efforts with an emphasis on climate resilience. As you can see, we have three folks in that project team joining us today to share more about that work. And I'm also joined remotely by my co-host, Ariana. Before we get started, as always, I've just got some housekeeping stuff. So an introduction to the reserve system, the Science Collaborative, and this series. So the National Estuarine Research Reserve System is a national network of 30 unique research reserves as shown on this map. More on the way. It's a NOAA program that works in collaboration with a local place-based partner, either a state agency, university, or nonprofit at each site. Each reserve site includes programs focused on the activities you can see on the slide there, which include land stewardship, research, and scientific monitoring, training programs for the public and local officials, and education. Science Collaborative supports science for estuarine and coastal decision makers by coordinating regular funding opportunities and supporting user-driven collaborative research and transfer activities that address critical coastal management needs identified by the reserves. The Collaborative Science for Estuary series features project teams supported by the Neuroscience Collaborative, program staff, and partners. Speakers share their unique approaches to addressing current coastal and estuary management issues. And if that sounds interesting to you, please feel free to join us for future webinars to learn more about the methods that are being used in these projects and how you might integrate them into your own work. A quick note on informed consent. Webinars are a valuable learning opportunity for our program. Insights from speakers and participants like you can help inform how we support others in doing collaborative science, for example, in the development of future workshops or webinars, or resources such as tools, tips, and case studies to add to our guide to collaborative science, which is open access. With that in mind, we just want to inform attendees that any insights or feedback that you share, for example, in the chat or via the webinar exit survey, may be used for program learning and development. Please note that if we do capture a direct quote from you that we'd like to use for learning purposes, we will reach out to you beforehand to seek permission and provide an explanation of its use. All quotes will be used anonymously, and we're grateful for the opportunity to learn alongside all of you. Final note here on housekeeping, all attendees are muted upon entry. We'll be handling the presentation-related questions via Zoom's Q&A feature or the chat. You can pop questions in. We'll be monitoring both places once we get there at the end of the presentation. You can enter questions for presenters as they occur to you throughout the session, and we'll plan to discuss as many as we can following the presentation today. Chat is visible to all, so you can use it to chat amongst yourselves or to ask organizers and hosts technical questions. And with that, I'd like to introduce our speakers for today. So joining me are Jesse Jarvis, a professor of biology and marine biology at the University of North Carolina, Wilmington, Stephanie Kamel, a professor of biology and marine biology at the University of North Carolina, Wilmington, and Aaron Shields, a research scientist for the Chesapeake Bay National Estuarine Research Reserve in Virginia.
All right. So we have a lot we want to share with everybody today. So I'm just going to go ahead and get right into it. With this project, we had a really excellent collaborative team, and you're going to get to hear from three of our core team members today, myself, Aaron, and Stephanie. But we would be remiss if we didn't mention Circe Gonzalez, who is our collaborative lead, who did an excellent job of coordinating all of our interactions with our end user work group, which we renamed the Savvy Resilience Brain Trust. And so Circe was great at not only keeping us moving and on task, but finding a way to keep everybody working together and contributing in a way that they were comfortable with, which is something we learned a lot about throughout this project. Our Resilience Brain Trust really has a wide different types of members within that group, and we were really lucky to have them to work with us throughout the entire process. And it really was truly a collaborative experience, and they made sure that all the products we're going to talk about today are things that can be used outside of our systems and anywhere you're trying to use eelgrass and try to restore eelgrass. And I think one of the reasons why we all work so well together is because we're all really focused on helping to conserve and manage and protect and even restore eelgrass. And this photo was taken by Aaron at the Chesapeake Bay, one of the reserve sites in the CB Nerva. And eelgrass in our systems provide really important ecological functions, including everything from sequestering carbon to improving water quality conditions, to also providing really important habitat for economically and ecologically important commercial and recreational fishery species. And seagrasses do this because they create habitat, right? They create cover at the bottom. Eelgrass is a marine flowering seagrass plant. We find them in a lot of our estuarine systems. And eelgrass in particular is important because it's found throughout the entirety of the northern hemisphere. One of the issues, though, is that eelgrass is a temperate species. And so as temperatures continue to warm, eelgrass is starting or continuing to decline in many systems. And so here's a figure representing some of the data from the Chesapeake Bay system right now. And that blue line is the proportion of total Chesapeake Bay aerial cover that's made up by eelgrass in the high salinity regions. And you can see that that line declines over time. It's declining right alongside this red line, which is the temperature data for the same system, which is going up over time. And as that red line increases above that sort of shaded area, which represents the physiological threshold for eelgrass dealing with high heat and high temperatures, we continue to see that big decline of eelgrass. Now, eelgrass in the Chesapeake Bay is not just declining because of temperature. There are many factors, including light. But in the last 20 years or so, one of the big driving factors has been increased temperature stress. Now, as soon as we've started noticing that eelgrass was declining, and this is something that, again, is not just happening in Chesapeake Bay, it's happening in many systems throughout the species distribution. But as soon as that started happening, seagrass ecologists, coastal managers started looking to ways to actually restore seagrasses. And we found lots of different methods to do this. We would take whole shoots and transplant them from one meadow to another. We would literally nail shoots into the ground. We would tie shoots onto burlap. We would just throw seeds off a boat. It's called broadcasting, make it sound fancy. We would also inject seeds into the sediment. All of these were different ways we tried to actually restore seagrasses and eelgrass in particular. And there's no one method that really works everywhere. It's really site specific for what would work for your system. In Virginia and in North Carolina, we find that seed-based restoration tends to be the most effective. And in particular, broadcasting seeds tends to work really well in our system. So for the rest of our presentation, that's what we're really going to be focusing on. Now, because we've been able to do seagrass restoration for a long period of time, we've actually had a chance as a global community to kind of look and see how effective we're being when it comes to our restoration. And unfortunately, seagrass restoration, in particular eelgrass restoration, is not all that successful. In fact, after about 2 years, only about 22 to 44% of seedlings survive on average globally following a restoration effort. And so this is really not something that if you're a coastal manager or if you're a decision maker, or if you're trying to make a vulnerability assessment, this is not the kind of information you want to see, right? You would like to see much higher numbers when it comes to restoration success to make some of these really hard decisions that coastal decision makers have to make. But there is some good news from this. The number isn't zero. There are some spots that had really high success rates, including in the Virginia coastal bays, right? And when you look at these sites that actually were successful when it comes to restoration, there are 3 things they have in common. The first one is careful site selection. So putting out a site where seagrasses can survive currently is key. The second one is using large numbers of seeds and shoots, and that kind of helps overcome any kind of unexpected incidents that you may not be prepared for with restoration, things like grazers coming in. And the last one, and the key one, is removal of the stressor, right? And that works really well when you're doing restoration in a system that maybe has potentially poor light conditions, because you can manage. It's not easy, but you can manage to reduce nutrients coming into the system or sediments coming into the system. But what do you do when the stress you're trying to restore is something like climate change and increased temperatures, like what we're seeing in the Chesapeake Bay in Virginia the last several decades? Now, thankfully, we're not the only ones that are asking these sort of questions. Our colleagues in terrestrial systems and in coral reef systems have been asking this for a while. And they found that when you are working with a stressor that you can't remove before restoration, one of the things you need to try to maybe think about doing is trying to make your target population more adapted to that stressor. And one of the ways to do this is through assisted migration. The method of assisted migration we're focused on in this project is seed migration. And this is when you have seed sources that you move climatically or geographically within their current ranges. So we're not introducing new species. We're taking eelgrass from within its current distribution, which along the west coast of the Atlantic Ocean is from North Carolina to Maine, and we're moving, hopefully, thermally adapted populations from one area to our area of interest, which in this case would be from thermally adapted populations, moving those into Virginia with the hope that if we could do restoration with those populations that have already adapted to that temperature stress, they're more likely to survive and will increase our overall restoration success rates. Now, to be able to do this, we need to find thermally adapted populations. So I'm going to make the case that we think North Carolina is a great source for that. All right, and there's a couple of reasons. First off, you have to have populations for this restoration that are able to both withstand that stressor, so be able to survive under higher temperatures, and then also, if there is a dieback due to that stressor, can recover quickly, right? And so in North Carolina, we have our eelgrass is exposed to high temperatures for a very long period of time every summer. And so this figure here is showing you the number of days each summer the eelgrass is exposed to stressful water temperatures in both North Carolina, which is the red part of the figure, and in Virginia, which is the blue part. And you can see that obviously North Carolina has more days that are hotter. We're further south, that makes sense, right? But what's really interesting is that because we had data that we can go back to the 1960s with, we can see that, you know, in 1961, on average, eelgrass in North Carolina experienced 84 days of stressful temperatures in the summer. By 2019, that had increased to 156 days. And so our eelgrass is experiencing high temperatures for a much longer period of time, and that has gradually increased over the last 6 decades. And so that's something that's given them potentially a chance to adapt. So that's the duration of the stressor. Our plants are experiencing that, and they are experiencing it for longer periods of time than Virginia. What about the magnitude of the stressor? What about high temperatures? Well, this figure is showing you daily mean water temperatures for just the summer months in one of our Sentinel, North Carolina monitoring sites. And so these triangles represent daily values. They're not the 15-minute data. We've already averaged all that. So that is a data for each day. Any of those triangles that are above that dotted line are when eelgrass in North Carolina in the summer is experiencing stressful conditions. It's the same area as that shaded line in that first figure. Anytime they're above the dashed line, that means that eelgrass in North Carolina is experiencing temperatures that have been documented to cause declines in places like Virginia. And they're experiencing on an average daily temperature in North Carolina. And anything above the black line are temperatures which have been shown to be lethal. And so you can see that, and this data is from 2009 to 2019. And so our eelgrass has been experiencing chronically stressful water temperature conditions in North Carolina over a very long period of time, and we still have eelgrass in our system. So we'd like to argue that our eelgrass can withstand and be resistant to high temperatures. Now, that doesn't mean we don't see decline. In fact, we see declines in eelgrass every year. And so here's a drone image of one of our Sentinel monitoring sites we took in June of 2018. And then we took the same imagery of that same spot, but in September of the same year. And you can see that our meadow area had declined by about 80% during this time period. And all of that loss can be attributed to loss of Zostera from the system. Anything that's still left, the dark area that's still left in that picture is Haliduli. It's our tropical species, which kind of doesn't mind the hot temperatures, so it hangs on. What's interesting is that this same decline had happened in Chesapeake Bay, and it has happened in Chesapeake Bay multiple times. It would take several years for that to recover. It would take a long time for us to get back to the same kind of image you see in June of 2018. We went back out to that same site in June 2019, and it was the exact same cover as it had been the previous year. And so our meadows, because they have shifted the life history strategy to rely on seeds to recover on an annual basis, have a really great mechanism to recover from a thermal stress. So North Carolina populations seem to be able to resist high temperatures at temperatures higher than other populations, and they have a good recovery mechanism, which means they are potentially good as a donor, maybe thermally resistant donor population for Virginia. So can we restore seagrass to be climate resilient was our big question in this project, and we had to ask two underlying questions to really get to that big one. The first is, are we actually finding thermally adapted eelgrass in North Carolina? And if we are finding that, can we demonstrate that they can actually do better in Virginia? And we did that with a fully reciprocal common garden experiment that we're going to talk about in the rest later in the presentation. So getting to that first question, why are North Carolina eelgrass beds more resilient to temperature stress than Virginia beds? I'm going to pass it on to Stephanie to answer those.
All right, good afternoon, everyone. Before I begin, I just need to mention that I am currently a program officer at the National Science Foundation. So the views and opinions expressed here today are my own and do not reflect the views and opinions of the National Science Foundation. So as Jesse said, part of my responsibility in this project was to ask the question of why are seagrass in North Carolina more resilient to temperature than Virginia beds, and we wanted to approach this from a genomic perspective. And so as you all know, most organisms, including seagrasses, have double-stranded DNA, which is transcribed to messenger RNA, which in turn is translated to protein, and those proteins are responsible for various functions and phenotypes of a given organism. And so every so often, a mutation arises in that DNA that gets carried through to the messenger RNA, and when translation occurs, we end up with a new protein, and that protein can then contribute to a novel function or a novel phenotype. And this is important because this process forms the raw material for evolution by natural selection, and it's a really important way by which populations adapt to changing environments. And so we really wanted to ask the question of, could we detect these important variations in the Zostera genome, and were these variations related to temperature? That has been the major stressor that we've been thinking about throughout this project. And one of the reasons why we can do this is that Zostera has a functionally annotated genome. And so we can go out and we can sequence our Zostera in North Carolina and Virginia, and we can compare it to that genome to try to identify specific regions that might be under adaptive selection, and then again try to ask the question of if those regions have any particular function and how that might be related to temperature stress. So in order to do this, we went to Virginia, and I say we, I don't go on boats, I don't sample things. This is all Jesse and Aaron and their colleagues and collaborators who went to Virginia and North Carolina, sampled 10 meadows in each state, collected between 10 and 12 shoots per meadow. Those shoots were sequenced. We targeted 10 to 20 sequence and coverage, and we sampled these meadows in both May and September with the goal of trying to look at genomic composition in plants before and after that summer stressful season. I'll also say that we tried to sample at least 5 meters apart to avoid detecting clones, because we wanted to maximize, obviously, our genomic coverage. And so just a few things I want to say about these sites. First off, genetically, they're pretty different. So as you can see, this is just a simple assignment plot, and all I want you to note is that North Carolina is clusters blue and Virginia clusters green, which suggests that there's pretty limited gene flow between these regions. And so any kind of adaptation or evolution within these regions is likely occurring independently. Environmentally, as Jesse's already mentioned, they're also pretty different. And so these are just density plots depicting a variety of temperature metrics. We have mean temp, max, HGH are heating degree hours, and then at the bottom there you see percent time above 28 degrees Celsius. North Carolina is pink, Virginia is blue, and what I really want you to just take home is that it is hot in North Carolina. And so the plants in North Carolina are evolving independently from plants in Virginia, and they're evolving under hotter temperatures and greater heat stress than the plants in Virginia. And so the first thing we wanted to do after we collected our environmental data and our genomic data was ask the question of, are there significant associations between regions in the genome and temperature? And I apologize for this figure. It's not great, but I'm going to take a little bit of time and walk you through this. So what I'm showing you here are a series of what we call Manhattan plots. They're so named because apparently they look like the Manhattan skyline. And what they are depicting are associations between regions in the genome and various temperature metrics. So as you can see on the X here, this is position in the genome. Zostera has 6 chromosomes, and so you can see that with the different colors. Here's chromosome 1 in gray, and then you have 2, 3, 4, 5, and 6. On the Y-axis, you have a number of different temperature metrics. One of the things we did was bin them by categories, so mean, variability, so coefficient of variation, standard deviation, and extreme, so max temp, number of prominent warming events, max heating degree hours, things like that, to just ask if maybe seagrasses were responding differently to different types of temperature stress. And then what you're looking at is this sort of association analysis where regions of the genome that are strongly associated with mean temperature are kind of indicated by these long lines, right? So each dot above this dashed red line indicates a significant association between a genomic region and temperature, and kind of the bigger the line or the higher the line, the more significant the association. So as I said, we collected samples in May and September. What I'm showing you here are September data because the signal was stronger in September, which makes sense because what we were measuring in September were the plants that actually made it through that stressful event. Another thing to note is the comparison between Virginia and North Carolina. The pattern is much stronger in Virginia. Those associations, there seem to be more of them, and they seem to be stronger in Virginia than in North Carolina in September. And I'm going to get back to that in a minute, but I just wanted to make that note. But what all of this is saying is that for a range of different kinds of temperatures, we are finding significant associations between genomic regions and those temperature metrics in both Virginia and North Carolina. So this idea is that already we're seeing potential adaptive responses in the genome in response to temperature stress. So this is another way, again, I think you can call my part of the talk horribly complicated figures that don't really help you understand things, but I'm trying my best. So this is another way of visualizing these associations between genomic regions and temperatures. And I want to point you to two main messages here. So one of the things we wanted to ask was, are there specific SNPs, regions in the genome, single nucleotide polymorphisms, that are associated with a particular temperature metric? Like maybe there's SNPs that are only responding to mean temperature, and maybe there are some that are only responding to extreme temperatures. And so the short answer for that is that that's not the case. If you look down here, these are all the different kinds of categories of temperature. So blue is mean, yellow is variability, red is extreme, and green is duration. And what you can see is that this SNP here is found in all of these different association analyses. And you can see that across every single SNP. And so what this is saying is that these regions of the genome are responding to temperature generally. And we don't seem to be seeing extreme temperature, genomic regions or mean temperature genomic regions. So that's one thing I just wanted to note. The second, and I think more important thing, is when you look at the comparison between Virginia at the top, so here are Virginia temperature metrics, and North Carolina at the bottom, what you see is that the SNPs do not overlap, which means that the regions of the genome that are associated with temperature are different between Virginia and North Carolina, right? So these regions of the genome are implicated in temperature in Virginia, and they are not found in North Carolina. In North Carolina, it's these regions of the genome that are implicated in temperature. And so this is important for a couple of reasons. Number one, it means that there is no one way to build a resilient seagrass. And so this idea that there's this one super genotype out there that's going to solve all of our problems is not the case. But on the other hand, it means that the opportunity to potentially find thermally adaptive genotypes has gotten much broader because there are different molecular pathways by which you can achieve, if you will, thermal resilience. And that there are different molecular pathways that are adapting to different types of temperature stress. So I think that means that there's just a lot of possibility for going out and finding thermally adapted genotypes. And that's just what I wanted to show. And it also does mean that there isn't that idea of parallel evolution, that these different populations are converging on that same genotype. They're converging on the same phenotype of thermal resilience, but they're doing it a different way. The next thing we want to ask is this idea, because ultimately we were really interested in if we could distinguish thermally resilient populations within our sites. And so essentially, we were wanting to know is, could we find sort of hot adapted versus, cold adapted or not hot adapted meadows? And so this is just going back to the environmental data, because before we could actually ask about genetic comparisons, we had to ask the question of, do these things even vary environmentally? And so we threw our environmental variables into a PCA, and we found that in both sites, or in both states, our meadows do actually segregate out into hot, indicated in red, and cold meadows. And so both within North Carolina and Virginia, we're finding these meadows that we're calling hot and these meadows that we're calling cold because they actually do have different temperature profiles. And so we now wanted to ask, okay, these things seem to differ thermally. Do they differ genetically? Are we seeing genetic differentiation between these thermally different temperatures? And so the answer is yes. I'm going to show you yet another figure, and I'll walk you through that to kind of explain, because there's two parts to this. One is, are these meadows genetically differentiated? And does that genetic differentiation relate to the SNPs that we found to be associated with temperature, right? We really want to ask, is temperature driving this genetic differentiation? Okay, so here's this figure. I've separated into four. We have blue, those are our mean temperature categories. Yellow is variability. Red are our extreme temperatures. And green is duration, right? How long they're subjected to hot temperatures. So for the first part of the question, is there differentiation between hot and cold sites? And again, I'm showing you Virginia, and I'll explain why in a minute. The answer is this analysis here, and the answer is yes. Between hot sites in Virginia and cold sites in Virginia, there are 2,882 regions of the genome that are different. Great. Now we want to ask, do any of those regions of the genome relate to temperature? And that's where all these overlaps come in. And the answer is yes, that there are a number of SNPs that are contributing to differentiation that are related to temperature. So we have SNPs that are related to median temperature, mean temperature, standard deviation of temperature, and all of this kind of stuff. So at the end of the day, we're able to detect significant regions of the genome that are related to temperature that are contributing to the divergence that we see between meadows. So we're able to say with pretty good confidence that we can detect the underlying genomic differentiation between our hot and our cold meadows. And I just want to show you one more figure to kind of think about that, is we've actually plotted different allele frequencies in our hot, which are the top two meadows, and our cold meadows. And we're seeing that the distribution of allele frequencies is different, right? The hot meadows have a distribution of allele frequencies here and here and here that are very different from our cold meadows. And we can actually ask, what are these allele frequencies? What are these SNPs related to? And in a number of cases, we're finding that they're related to specific genes that affect things like metabolism and growth. And so we're able now to finally understand from a functional perspective, what changes are occurring in the genome that are enabling resilience to temperature. Now, I will say I've been talking a lot about Virginia, and I want to say this because all is not lost for Virginia. There is adaptation in response to thermal stress that is occurring in Virginia, right? They are responding adaptively to temperature. The question remains, are they going to be able to respond quickly enough to the rapid changes in temperatures, or might they need help from genotypes that have already had a much longer history evolving under these extreme temperatures? And that brings us to North Carolina. So we have our environmentally different meadows. We wanted to ask the question, are they genetically different? The problem is that in September, we actually saw mortality in North Carolina. And the mortality looks like this. And so what has happened was that the meadows that didn't persist were the meadows that were primarily our cold meadows. And so this comparison between hot and cold in September wasn't really able to happen. And so what we compared in September was actually mostly sites within the hot. And so it's really neat that we actually saw these associated SNPs with temperature within the hot, because that suggests that within North Carolina, adaptation is also occurring at a very fine thermal scale, and that we're seeing continued adaptation in response to these more extreme events that we're seeing in North Carolina. So our thermally resistant North Carolina seagrasses are continuing to adapt to these changing temperatures. So how do we ask the question of, do our hot and cold meadows vary when our cold meadows are essentially all gone? Well, we can go back to our May data, where we have a complete set of genotypes, and we can actually ask the question, in May, can we compare our non-persistent meadows to our persistent meadows, and can we see a difference? And the answer is yes. We found significant genetic differentiation between persistent and non-persistent meadows. So, which indicates that not only is North Carolina evolving thermally resilient genotypes, but they are also able to evolve genotypes that are able to kind of resist, in a sense, that temperature-associated mortality. So there's a lot of really interesting adaptation that goes on, And this is why North Carolina seems to be a potentially really important seed source, because they have evolved a number of adaptive responses in the face of pretty strong thermal stress. And so to sort of answer the initial question that we posed, North Carolina meadows are more resilient because they possess unique genetic variants that confer the ability to withstand extreme heat stress. And I think that at the end of the day, that was the most important take-home message, is that there are really key regions of the genome found in North Carolina seagrasses that have enabled them to persist under these highly extreme thermal regimes. And I think I will turn it over to Erin now.
The second research question we were investigating was, will these North Carolina eelgrass perform better than Virginia plants under stressful water temperatures, regardless of location? And so to investigate this, we looked at, as Stephanie mentioned, we looked at these 10 populations within Virginia and 10 within North Carolina. And we implemented a fully reciprocal transplant experiment. And so within these populations, we did this back in 2023, we were looking at various metrics such as genomics, as Stephanie just talked about. We were also looking at reproductive effort, percent cover of the eelgrass beds, shoot density counts. And we also harvested the reproductive shoots in order to get their seeds for our transplant experiment. And all of these methods are available in a SOP that was developed through this project, and you can find on our project page. Essentially, you go out to the meadows, and in our area, it's typically early May when the eelgrass is producing these reproductive shoots and the seeds are ready. And so you harvest the reproductive shoots, you bring them back to, a greenhouse or some kind of place that has tanks where you can put them in recirculating or flow through tanks. And then after a couple of weeks, the seeds fall out of the shoots, and then we filter everything through a sieve where the seeds are maintained and everything else you can get rid of. And so once we got all of our seeds, I drove them down to North Carolina, and so they were all the seeds from all 20 meadows were stored together in the same conditions at the UNC Wilmington facility. And so from these populations, we selected 3 per state that we wanted to use for the transplant experiment. And this was based on different resiliency metrics. So we chose 3 populations that scored high in reproductive output. They had a lot of viable seeds. They had relatively high percent cover and shoot density count, so a healthy meadow. And so we took the seeds from these specific 3 populations per state, and we implemented this fully reciprocal common garden in both North Carolina and Virginia. So every seed source was planted at every site. And before the seeds go into the water for the experiment, they're soaked in a copper sulfide solution, and that's to prevent any potential pathogen, harmful pathogen exchange between the states. All right, so we're looking to build resiliency into already existing beds, right? We didn't want to set up this experiment in bare areas that hadn't had any eelgrass growing for, we wanted to see success and to see if we could build this resilience into already existing beds. And so the plots were placed within these meadows we were already studying in bare areas. So bare areas within the meadows are just adjacent to the meadows where we know that eelgrass can grow. And then it was a randomized location of treatment. So the treatment being seed source, where the seeds were coming from. And we placed 150 seeds into the 1 meter middle section of each plot, and they are just kind of, these seeds sink rapidly, and you just kind of, for this, we patted them into the top of the sediment. And we left at least one empty cell in between all restoration plots just to prevent, growth from the neighboring plot. And these were marked with floats or GPS coordinates so that when we were monitoring, we could go back to the exact same spot and know that we're monitoring that specific seed source. So one of the first things we wanted to look at once we started monitoring the success of these plots was this idea of home site advantage. So are we seeing plants performing better at their own site where they were sourced from compared to being moved to other sites? So this graph is only showing, you might notice it's only showing 4 sites, whereas I said we did 3 in each state. That's because our Virginia 5 site actually had an excellent year with eelgrass, and all the surrounding bed overgrew our plot, so we weren't able to distinguish, what was from what we planted and what was from the surrounding bed. And then the North Carolina 2 site got had a sandbar move over it and had no success. So here we're focusing on the 4 sites where we did get seedling germination. And so each of these plots is a different seed source. And then the y-axis is just the eelgrass shoot count, so the density. And then the x-axis is transplant site, so where the seeds were put in. Blue is the Virginia sites and green is the North Carolina sites. And so what was interesting is we actually only found one instance of home site advantage, and that is this Virginia 3 site, where the seeds from Virginia 3 performed the best planted at Virginia 3 compared to any of the other sites. And then we looked at, well, is there variation in how these seeds are performing across season, and does it depend on where the seeds were sourced? And so this graph is set up differently. So each graph now is your transplant site. So Virginia 1 on the top left, Virginia 3 on the top right, and then your 2 North Carolina sites on the bottom. Again, the y-axis is shoot count. And then this time the x-axis is seed source. And now we're including Virginia 5 and North Carolina 2, because while we can't look at how these seeds sourced from there performed at their own site, we can see how they performed at all the other sites. And then the different variations of blue are just binned into season. So we have our lightest blue is spring, and then early summer, and then the dark is late summer. And so in Virginia 1 on the top left, we did see early season success across most of these seed sources. However, we saw declines in the late summer and not a whole lot of survival by the end of the summer there, and it didn't really differ by seed source. And we had a similar story with the North Carolina sites on the bottom, where we did see early season success. We got germination across all seed sources planted there. However, very little to no survival after the stressful summer period by August and September. However, we saw some interesting results with Virginia 3, and that's on the top right. And if you remember, that's also the site that we saw the home site advantage there. But here we saw North Carolina seeds doing very well, particularly from this North Carolina 1, North Carolina 2 site. We saw early season success, but we also saw it persist and even increase throughout the summer and have really high numbers after the stressful summer period. Well, so what did the temperature look like at these sites? Here we plotted cumulative hot degree hours per site over time. So this is the hours they're spending above this 28.6 degree threshold that has been documented to be kind of the stress threshold for eelgrass in this area that Stephanie talked about earlier. And so North Carolina's hotter, right? We know that, but these two North Carolina sites are acting very similarly, whereas the Virginia sites are a little bit different. And this Virginia 3 site is actually the coolest. And again, that's the site that we found the home site advantage with their seeds. And we also found North Carolina plants to do quite well there. All right, so I want to emphasize that this is 1 year of post monitoring that we were able to do for this project. So what's next? So 2025 and beyond. Well, we're continuing to monitor these plots and through a variety of ways. This Virginia 3 site is actually one of our, it's our reserve site, Goodwin Islands. And so through the system-wide monitoring program, we already go out there to monitor the seagrass beds. And so we're able to kind of incorporate these restoration sites into our monitoring. And we are also fortunate to get placed with a Margaret A. Davidson Fellow, who is continuing some of this monitoring work and expanding on some of it through this field season. So that's exciting. And I've put this temperature graph on here. This, I want to just, last year when we were monitoring the transplant sites, it was not a particularly hot year for Virginia. I mean, every summer here is stressful for eelgrass. However, we didn't see any major like heat wave event causing mass die-offs throughout Virginia last year. This year is a different story. And so this graph is showing daily mean temperature. The red is from our system-wide monitoring program continuous station at this Virginia 3 site. So we have data back to 1998. So we're able to look at the historical trend. And then the blue dash line is just the upper 10% threshold based on the historical data. And the green is the daily mean from this year. And so we've had an extreme heat wave event, and it happened really early. And that can be particularly bad for eelgrass because June is usually the time where it's trying to store up its reserves to last the rest of the summer. But you'll see we had daily means up to 32 in June, which is really, really hot. And so just highlighting the importance of being able to continue this monitoring, you know, before and after this major heat wave event. That's going to be really interesting. All right, I'm going to pass it on to Jessie again.
Okay. So were we able to answer our question? Can you restore seagrass to be climate resilient? Well, I think that would be pretty cheeky to say we did in the 2-year study. So what I think we did do, however, was make some big strides when it came to identifying mechanisms about what could actually drive this and you do climate resilient restoration in the future. We found a lot of differences in both North Carolina and Virginia, and we found genomic variants that were linked to multiple different temperature metrics. So something that's key that kind of highlights we need to think about temperature in many different ways and maybe just the average temperature, for example. We did find that molecular pathways towards thermal resilience were different, and they differed by state, and that we're still seeing ongoing thermal selection in both Virginia and in North Carolina. And so we really shouldn't give up on either of those states. And that we also can identify and link those genomic variants to specific meadow types, hot versus cold. Which is something that's really exciting to be able to see because that really, again, reinforces this idea that we could have multiple different populations that could help with climate resilient restoration and not just necessarily thinking about getting one particular population. Like, don't everybody come to North Carolina. You may have some within your own sites, right? As far as the did also do a good proof of concept, I think, with some positive initial results for doing more climate resilient restoration. We did see some instances of North Carolina seeds outperforming Virginia, and we're really eager to see what would happen moving forward with that. And that seed source really is very important. And that's something we've kind of known anyway in the field, but now we need to think about it beyond just sort of genetic diversity, but also think about genomic function, right? And sort of like what's happening in more thermal resilience. And then finally, we need to do a little bit better job of matching seed sources to local temperature regimes and thinking outside of just, across multiple scales, including spatial and seasonal scales. I think that our initial results are really showing that these are things that are really going to have a big impact in the overall success of restoration going forward. And then any good research project always leads to more questions. This is something that we're really excited to look at our year 2 of monitoring, especially with this massive heat wave that they experienced in both Virginia and in North Carolina. What's nice is that we're going to continue to collaborate with our Savvy Brain Trust with several members of that group and continue with a project looking beyond Virginia and North Carolina and asking similar questions from eelgrass populations from North Carolina up to Maine. And this is funded by the National Park Service. We also have a lot of colleagues who are working not on this project, but sort of at the same time having similar conversations about their systems, and they're looking at within system or within state thermal differences and finding some really interesting results. And then lately, there's been a big push for some more bi-coastal collaboration. So looking at eelgrass resilience from both the Atlantic coast and the Pacific coast and making restoration more resilient by broadening our scope and our knowledge base. What's great about all of these projects moving forward is that they don't have to start from scratch, because of our project and the fact that our collaborators really pushed having products that could be applied outside of their system and they could be applied immediately. So this QR code is a link to our project page that the Science Collaborative put together for us, and it contains links to all the SOPs for the field trials, including a seed source site selection flow chart, the DNA extraction protocol, bioinformatics pipeline that tends to be the scariest part for a lot of folks, and that has been written out that you can plug your data into as well, and then including the R script for the workflow for the genetic analyses. Our data sets, the environmental and monitoring data, are currently available, and all genomic data for all 20 sites will be published after 6 months. We're going to get a couple papers out first, but that will all be available as well for anybody to use. So these are all the big products that are available right now on the website. So please, if you're curious, please look at that. And with that, we will say thank you, and we'll move over to the questions part of it. So,
Thanks, folks.
all right, so we'll get started. Someone asked, or I think I ran. Are you able to detect the rate of heat-related adaptations? And have you seen the change in the rate in these adaptations? So that would be for Steph, I think.
We haven't done any rate change work. There might be a way to do some sort of Markov chain coalescent analyses, but that's beyond the scope of this project. But I think it's doable. But to answer your question, I have not done it. Oh, I guess I think I saw a question about outbreeding depression from the chat. I'm not thinking about it at this point, in the sense that when you look at, so you can actually distinguish the populations looking at the structure analyses. When you do the PCA, there's not a ton of variation that's explained by the PCA. And so I feel like in terms of their evolutionary history, they're close enough in population size that outbreeding depression might not be a problem. That being said, that's something that we can monitor through our genomic monitoring of the plots, and something to consider. But a lot of this initial work was the proof of concept of, could we get the seeds? Could we transplant the seeds? Were we going to see germination? And that's kind of the initial sense. But those are all sort of important follow-up questions of, what are patterns of hybridization going to look like between North Carolina and Virginia plants and things like that? So yeah, it's a great question. We don't have any data to speak to the outbreeding depression at this point.
But that was another question they asked: were the differences in SNPs? Oh wait, is that what you just answered? No. You detected due to differences in underlying genotypes between plants, or differences in gene expression between population?
So we didn't do any gene expression analysis. So these were all of this stuff was looking at changes in the DNA sequence. So yes, so there's so much, and we talked about that. There's looking at the transcriptome, there's looking at the methylome, there's all of these subsequent information, ways that you can respond to changing environments. And so we really wanted to ask, at the very basic level of changes in DNA sequence, are we able to detect those potential adaptive genomic variants? And so we have, for this project, there's no gene expression work.
The next question is, how are you thinking about the application of these results in other regions? And do you expect to see similar patterns of genomic change in eelgrass in other regions? So I'm going to answer the first part, Steph, and then I'll give the second part to you. So we're thinking about we actually one of the documents we shared with our brain trust was the lessons learned, and so we learned a lot about how to do this, including a lot of steps that you can kind of just add one. You mentioned in just a quick 30 seconds, like we have the DNA extraction protocol. That took months to figure out with some of our messier or dirtier DNA. So we're taking a lot of lessons learned, but I think what we found is that this is and we were not the first ones to do common garden. We weren't the first ones to do this kind of restoration. But I think that we learned what was really important to monitor alongside with our actual plot data. It was really important to get data from the nearby meadows. It's really important to get information about sexual reproduction. Like that is something that we really have some key data on. And so those are the kind of things we're trying to share with our an application in other locations. And then, Steph, do you expect to see similar patterns of genomic change in eelgrass and other regions?
So that was the big question for the National Park Service grant was, were we going to be able to see, when we sort of sequence across, we have samples from Maine all the way to North Carolina, and are we going to see the same kinds of patterns? And so we don't have the environmental data for the NPS to do those environmental association analyses, but what we can do is the outflank analysis, just looking at patterns of divergence. And so even up in Massachusetts, we just did some quick comparisons between the northern populations and the southern populations. I want to say Winthrop maybe versus Tuckernuck Island. I can't totally remember. And we do see patterns of adaptive differentiation. And we used some kind of existing sea surface temperature data, and some of those patterns were driven by SNPs that were associated with those very coarse kind of sea surface temperature data. So even up in Massachusetts, where maybe temperature is not the most important thing, we are still seeing signals at this very early stage of temperature-driven divergence. So will the patterns be of the same magnitude? I don't know, but I can tell you we're already kind of seeing the initial, that there, I don't know about magnitude, but of kind of type, if you will, we are seeing that. Yeah. But stay tuned, because the NPS data is going to be pretty large scale and interesting.
Nick, I'm going to mess up here and go down your list a little bit. So this is, I think I'm going to throw this to Aaron. Have you seen eelgrass moving to deeper water in the lower Chesapeake Bay or lower York River, take advantage of the relatively cooler water in deeper areas where they can still get enough light?
In short, yes. I'm personally seeing it just in our transects that we measure. I know, the annual survey that's done here every year is seeing it. And it is very interesting in that light is getting better. The Goodwin Island site, the Virginia 3, we're able to monitor the light conditions there over a long period of time, and it's just getting better and better and improving. And so yes, our long-term transect there, we're seeing grass grow deeper than we have before. And so that'll be a really interesting thing to track through the annual survey, as well as maybe extending. The problem is our transects don't go that deep, and so maybe extending them deeper and being able to track that over time to see if that is kind of like becoming a colder water refuge in those deeper areas now that they can survive there because of the light. Yeah, great question. Thank you.
So the next one is, if you use North Carolina seeds to restore in Virginia, would you also be initiating a life cycle shift to more annual expression? And I would say I don't necessarily think that's guaranteed. For one thing, if we don't have a hot year, our eelgrass can persist. And in fact, it was a documented perennial up until early 2000s. So I do think I don't think at this point they're not dying off until there is that heat stress. So there is a possibility, right, that you have a greater chance of having the annual life history expressed in that system, which would, again, you have to think about. If you have that, then there's a part of the year where you may not have seagrass or SAV in the area. In North Carolina, that's not as big of a deal because we have haliduli that's able to maintain year round and maintain the meadows. And so that's something that It's by switching to an annual, and we actually don't know if it's a big deal or not. We're still trying to figure this out, as Zostera declines, what's happening, but you don't necessarily have that redundancy. There's rupia in Chesapeake Bay, but it's really not sure that if you switch from this, what's going to be the implications. But I wouldn't think it's guaranteed right off the bat. All right. Aaron, you've been playing with this data a lot. Are there morphological differences between North Carolina and Virginia, or between meadows in a given state? And did you see any plastic morphological responses after transplantation?
Okay, sorry, I'm trying to read that. Yeah, so we did take a suite of morphological samples. We were surprised at how well Virginia scored on a lot of areas compared to North Carolina from the 2023 data. We saw, huge reproductive output. A huge amount of energy being put into reproductive shoots and seeds, and a lot more viability was higher in Virginia. Higher shoot counts, I think, pretty much across the board compared to North Carolina. We don't have length comparisons, but yeah, in general, North Carolina was scoring Virginia was scoring really well on all these metrics compared to North Carolina. Part of that, I think we are having fantastic eelgrass years here. Ever since this project we started thinking about this project and writing the proposal, eelgrass started doing great, which is wonderful. We have not had a large scale heat die off since 2015. So we've got 10 years where these beds have been able to recover. We have improving light conditions, and so they're really happy right now. And I think that's showing in some of that morphological data as well. Yeah. So that's why I'm very interested to see what happens after this current heat wave, because unlike previous heat waves, we're starting from a much higher number now. Like the eelgrass is distribution is much greater, it's healthier, it's happier than it has been before the previous bad die-off 10 years ago. And so hopefully that will provide some kind of resilience to this summer that we're experiencing. Yeah, I don't know. Hopefully that answered some of that question.
Maybe we have time for one more question, Nick. All right. The finding that different genomic regions are implicated in thermal tolerance in the two sites is really interesting. Are there other areas with eelgrass in between the two sites, potential for isolation by distance gene exchange?
So there's, correct me if I'm wrong, Jesse, but there is, right? There's the seagrass beds in the Outer Banks, and there's other places in Virginia, because did we sample coastal bays for, I don't know my geography. I do know that the Outer Banks is definitely between Southeast North Carolina and Virginia. So there are different sites that we have actually sampled. And so when we start integrating, that's the kind of question. So the answer is I don't know. I don't know what the Outer Banks genomic picture looks like, which is one of the things on the list to kind of see what that transition area looks like. And so hopefully we'll have some answers about that soon.
We are at time. Do you feel like answering one more question?
One or more questions. I didn't want to, but I'll go ahead. Are there Atlantic marine wildlife species that improve the resilience of seagrass species or growth? Maybe manatees. They understand that sea otters improve seagrass growth in Pacific. So I don't know if we know that. I think there are examples where they could help with distribution of seeds. And I think we're learning more, but I don't know. What do you think, Erin?
I wish we had manatees. I'd love to swim with them. I don't know. I mean, I haven't even thought about that much.
Future project idea, somebody.
Yeah, exactly.
Yeah. Next steps for research. Okay, that's all the questions I think we have, and we're a little bit over right now, so apologies to everybody, and thank you to the speakers for staying over to answer that last question. If you do have another question that occurs to you after the session ends, you can feel free to either email it to me. My email's in the exit survey link as well. You can put it in the exit survey. That message will go out as soon as you close the session. It'll also go out tomorrow, same time. Basically, you'll get a reminder email from Zoom automatically that says, here's the materials from the webinar. You can look out for the recording in the next couple days. Usually only takes us like as long as 2 days to get it processed and posted. So look out for that. And feel free to join us for future webinars. But again, just a big thank you to the presenters for today for putting together such a great presentation and for explaining things so well. All of us on the side were kind of like just chatting like the program officers about how good a job you're doing of presenting such complex information. So kudos. That was really cool. And with that, just again, big thank you, and I hope everyone enjoys the rest of their day.